Hello, good morning. My name is Dr. Rajvir Singh. I am Professor and Head of the Department of Microbiology in the Dental College ITS, Murad, Nagar, Ghaziabad. Today, our topic of discussion is anaerobic culture. In the anaerobic culture discussion, we will take it in two parts. In the first part, I will be talking about some theory of anaerobic bacteria and anaerobic culture, and in the second part I'll try and demonstrate the various ways of doing anaerobiosis by Macintosh Fildes jar and the gas pack jar. So this will be our endeavor in this topic today. Let us start with the topic. As I've told you, the topic today is anaerobic culture. First of all, we must know what anaerobes are. Anaerobes are the bacteria which grow in the absence of oxygen. Even the small amount of oxygen may prevent the growth and multiplication of these bacteria. These bacteria which require no oxygen at all and which die in the presence of oxygen, they are called obligate anaerobes. In fact, if we talk about the oxygen requirement, on the basis of this, we can divide bacteria into many categories. Like, obligate anaerobes grow only in the absence of oxygen, and they can be destroyed even by the oxygen presence only in 10 minutes time. Another type of bacteria are microaerophilic. As the name suggests, micro means small, aerophilic means oxygen liking. So these are the bacteria which grow best in the presence of 5% oxygen. Another variety is aerotolerant bacteria which grow best in the presence of 20% oxygen. And lastly, facultative anaerobes are the bacteria which can grow in all sorts of oxygen atmospheres. This here picture shows that various types of bacteria, how do they grow? We have taken here a deep stab culture. This is the tube which contains this here is the agar, nutrient agar. And it is in this we stab this with the bacteria deeply until this part. This part of the agar material, it has the lowest content of oxygen, whereas the top part here, this is the high content of oxygen. So depending on the amount of oxygen, various types of bacteria will be grown at various depths of this deep agar. See here, this is the obligate aerobes, meaning those bacteria which require oxygen. And this is shown here that these bacteria will grow at the top of the agar, deep agar, because here at the top is there is the highest concentration of oxygen. In this tube, it is the contrast of the first one. What is happening over here is that those bacteria which are deepest at the bottom of the tube, they will grow here because there is no oxygen, so these bacteria will be obligate anaerobes. These are the three other types. In this, these are the facultative anaerobes, meaning they will grow in the presence of oxygen and also in the various concentrations of oxygen. Aerotolerant are also this type. All these facultative anaerobes, they are going more on the high oxygen content, but these aerotolerant, they will be growing in the same amount in the whole of the depth of the agar. Whereas these microaerophils, they will grow slightly below the surface of the deep agar, showing that they require little amount of oxygen. Now, the question comes, when do you suspect anaerobic infection in a specimen? There are certain clues which may make you believe that this particular sample, this particular patient may be having anaerobic infection. First thing is, if the discharge in the wound or anywhere, it is very foul smelling, it is always suspect anaerobic bacteria. Then necrotic areas, the areas which are dead, the tissues which are dead, they are likely to have anaerobic growth in them. And gangrenous areas also have the presence of anaerobic bacteria. Also, if there is crepitation in the tissue where the infection is there, then crepitation means there is gas underneath the skin, and it, like the bubbles, they burst in the form of crepitation. If this thing is present in the patient, always suspect anaerobic bacteria. Then black color, black discoloration of any discharge, that shows that the person may be having this anaerobic infection. Sometimes there are certain granules present in the pus, and these granules are very tiny, white to yellow color granules, and these granules are actually colonies of certain bacteria, and usually either nocardia or actinomycetes. Actinomycetes, as you know, is an anaerobic bacterium, and presence of these sulfur granules shows that the infection may be because of actinomyces. Certain samples, like blood samples, should always be cultured for anaerobic bacteria. There are certain precautions one must take while we want to do the anaerobic culture. First thing is, all anaerobic conditions must be maintained during collection, transportation of the specimen, processing of the specimen, and identification of these bacteria. Proper collection and transport of the specimens should be taken, and very important thing is immediate delivery of the specimen. It is very crucial. If there is delay and you anticipate some delay, then anaerobic transport medium should be used, where you should use the specimen to send to the laboratory. There are certain types of infections where one must always do the anaerobic culture, and these include abscesses, deep-seated abscesses, bites by animals and even human beings, blood, cerebrospinal fluid, exudates from various fluids, deep wounds, and dead tissues. If you have any of these, always, along with the aerobic culture, one must always conduct anaerobic culture also. Another thing to remember is there are certain specimens which are not at all suitable for anaerobic culture, and these are the specimens from places, from the sites of the body which normally contain anaerobic bacteria. For example, mouth, intestine. All these, if you want to culture feces, you don't have to do anaerobic culture. Similarly, sputum, throat swab, rectal swab, or any other thing which is including the saliva, they should not be cultured anaerobically, because in these the anaerobic bacteria are normally present. So it will be of no use to do these things. Now we come to how we collect the specimen. The best way to collect a specimen for anaerobic culture is by syringe and needle. And how do you do it? First, disinfect the skin with 70% alcohol and allow it to dry. Then aspirate the specimen directly into the syringe. Remove the syringe from the area and aseptically transfer the aspirated material into an anaerobic transport vial for fluids. If suppose there are times when you may have a dry tap, meaning that you cannot aspirate anything from the site, and you are unable to take out the specimen, then what you do is with the 0.9% saline, it is injected into the aspiration site, and then aspiration is done. In case of deep wounds, a deep swab is taken, and it should be always, the swab should always be transported in the transport medium, anaerobic transport medium. Tissue samples and biopsies are also good material for the anaerobic culture. Now, how do we transport the specimens? Like I told you before, it is very important to transport. Transport should be done immediately, and if there is a delay, do not refrigerate, do not try and refrigerate the specimen, allow it to stand there at the room temperature. If the amount of the fluid is very small, like less than 1 milliliter, then you should send the sample within 10 minutes of the aspiration. Small volumes, that is specimens from 1 to 2 milliliter, they are to be transported within 2 to 3 hours directly into by the syringe. But if there is large amount of specimen, then this fluid should be transferred to a sterile screw cap container at the bedside, and then it can be transported, and it should be transported between 2 to 3 hours. So these are the transport conditions for the specimens for anaerobic culture. There are various methods. After the specimen comes to the laboratory, it has to be processed anaerobically. And for anaerobic environment to be maintained, there are various ways. Number one is producing a vacuum. If we produce a vacuum, we remove air from the container, and in that atmosphere we can put the various petri dishes inoculated with this specimen and allow them to grow. Then the other method is oxygen displacement. That is, you displace the oxygen, you remove oxygen by putting some other gas like hydrogen or helium inside the cylinder, so that oxygen is displaced and in its place some other gas is taken out. The third way is oxygen absorption. It is done by various ways. You absorb oxygen and remove it from the atmosphere. Fourth way is by reducing agents. There are certain agents which cause reduction of oxygen, and there are certain chemical substances like glucose, maybe cysteine, ascorbic acid. All these methods are there for reducing agents. In a normal laboratory, we use two reducing agents, that is cooked meat in the Robertson's cooked meat medium, and the second is thioglycolate medium. So these are the reducing agents, and the fifth one is anaerobic chambers. In advanced centers, we have anaerobic chambers where inside the chamber there is actually no oxygen at all, and you put your hands into the chamber through glove areas, and that's how you do the anaerobic cultures. Same thing. So by reducing substances, I told you Robertson's cooked meat medium, thioglycolate broth, and another method is do the culture away from the oxygen, that is by deep agar tubes. The chemical exclusion is done by vacuum in the gas pack jar, and mechanical exclusion by vacuum is done in anaerobic incubator or by Macintosh Fildis jar. This is the Macintosh Fildis jar. I shall be talking about this in the practical aspect, where I will demonstrate how this Macintosh filled jar works. This is the jar, and this is a metal jar having a lid which is made airtight, and it has got one inlet and one outlet and a pressure gauge to show there is no gas inside, oxygen inside, air inside. And this the working of this jar I will be discussing in the demonstration part. In the gas pack principle, where we displace oxygen by various gases like hydrogen and carbon dioxide, in this particular case we use displacement by hydrogen and carbon dioxide. What is done here is hydrogen combines with oxygen in the presence of a catalyst, palladium or some other catalyst, and water is formed. So there is no oxygen inside the chamber. This is the gas pack which we are using. This gas pack contains certain chemicals which when you add water into it, it produces carbon dioxide and hydrogen. And in the presence of this this catalyst here, that is palladium catalyst, the hydrogen combines with oxygen to make water, and there is no oxygen left inside. So all these plates which are kept inside the gas pack jar, they are inoculated with the specimen, and if the anaerobic bacteria are there, they have the suitable environment anaerobic environment to grow and multiply and make colonies. This is the same thing which has been shown again. Reducing oxygen is done by various chemicals methods. These are glucose, thioglycolate, cooked meat pieces, cysteine and ascorbic acid. In routine laboratories, we use thioglycolate medium and the cooked meat pieces or cooked meat broth or Robertson's cooked meat medium for the as reducing agents for the growth of anaerobic bacteria. This is Robertson's cooked meat medium. These are the meat pieces in the broth, and this is the growth which has come inside the here. Same things has been shown here in this other picture. This is the Robertson's cooked meat broths. This is the deep agar culture. What we do here is that we have a tube in which nutrient agar is placed, and it is having a big column like this. The organism which is to be grown, it is taken on the tip of this straight wire. This is the straight wire. This wire is called straight wire, straight inoculating wire as it is called. And at the tip of the wire we take the bacteria or the specimen which is containing these bacteria, and it is stabbed deep inside like this. It is stabbed deep inside. And if and this is then taken out, this straight wire is then taken out, and if the bacteria are strict aerobes, they will grow at the top here, like this. This is the growth of strict aerobes, and if the bacteria are anaerobes, they will grow at the bottom of the tube. So this is called the deep agar culture of anaerobic bacteria. This picture shows the various methods of one of the various methods, that is the chamber, that is anaerobic chamber for the growth of these bacteria. See how this person is standing outside. This is the chamber here. These are the various materials which are used for the culture. These are the gloves. The person does not enter the chamber, neither his hands, only hands go into the chamber through these gloves, and this is all airtight. The media usually used in a routine laboratory are two media, that is thioglycolate broth and the cooked meat broth or the Robertson's cooked meat medium. This is the thioglycolate broth. These are the tubes, whereas this is the bottle. This is again this thioglycolate broth containing the growth of the anaerobic bacteria. This is RCM, that is Robertson's cooked meat medium. This is the there are two types. We should not be going into the saccharolytic and proteolytic part. We will do it when we study Clostridium. After you have obtained the anaerobic growth of these bacteria, they are identified by various methods, various chemical tests and other things. And after their identification, usually the antibiotic sensitivity test is done on these isolated bacteria. But in case of anaerobic bacteria, it is not customary to do the antibiotic sensitivity testing. But when it is done mainly against penicillin, clindamycin, cefotaxime, metronidazole, carbamazepine and chlorphenicol. These are the 6 drugs mainly used for the antibiotic sensitivity testing. Here it is a flow chart showing how we do the anaerobic culture. First of all, specimen has been taken with all anaerobic conditions. This specimen is inoculated in 2 ways. One, we put it in either in the Robertson cooked meat medium or in the thioglycolate medium in order to have absolute anaerobic environment, or directly plated on the blood agar. The specimen can be directly inoculated on the blood agar plate also. After this blood agar plate is incubated into in the gas pack jar or in the Macintosh Field's jar. In case the specimen was inoculated in the Robertson's cooked medium, it is allowed to grow for about 24 hours, and after 24 hours or maybe 48 hours, when the turbidity occurs in the upper part of the medium, that is the broth, turbidity indicates that there is a growth in the medium. Once this turbidity occurs, we take out this some amount of fluid from here and subculture it on the blood agar plate. This blood agar plate is then put in the gas pack jar or in the anaerobic jar, and then then it is incubated for about 2 days. After 2 days time, the growth will come, and this growth is then identified by various tests, and then antibiotic sensitivity test is done. After that, the report is sent to the various departments where the specimen has come from. So this is, in nutshell, is the process by which we do the blood culture. This is all for today, and now in the second part I will be showing you how we do the anaerobic culture in case of a Macintosh Field's jar and also in gas pack jar. Also, I will be showing you the medium for anaerobic culture, that is Robertson's cooked meat medium. So let us go to the demonstration part. Thank you. Today's practical is anaerobic culture. Anaerobic culture. You know, there are certain bacteria which do not require the presence of oxygen for their growth. These are called anaerobic bacteria. Some anaerobic bacteria are such that even when they get exposed to even slight amount of oxygen, they may die. They will not grow. So, and these kind of organisms are called obligate anaerobes. Most of the organisms which we encounter in our daily life, they are facultative anaerobes. That is, these bacteria are capable of growing in the presence of oxygen as well as in the absence of oxygen. But we are not concerned about these bacteria today. We are only concerned about the anaerobic bacteria, the bacteria which do not require oxygen for their growth. And so we have to create anaerobic conditions for their growth. How do we do that? There are three methods. One is by vacuum. By vacuum. Number two, oxygen displacement. And number three is by reducing agents. The vacuum method is done in the Macintosh LDS jar. Oxygen displacement method is done by the gas pack jar. And reducing agents, there are many kinds of reducing agents which can be used, like ascorbic acid, where there could be glucose, even glucose, cysteine, thioglycolate. In our normal laboratories, we use cooked meat, and the medium is known as Robertson's cooked meat medium. So we will see these three methods, how we approach to them. Is put here, and with the help of a sterile loop, we spread the specimen like this. So this is similarly, there may be other specimens which have to be inoculated in various plates, and suppose these are the plates which have been inoculated for anaerobic culture. We put them inside this basket. Once they are put, we put this inside this like this. We put the lid on top and clamp it. It is tightly clamped like this. Now, this one of the jobs, which is exhaust, which is the outlet, we put the rubber or the tubing of the vacuum pump to this and put the vacuum pump on. This should be closed and this should be open, so that all the air which is inside, it is taken out with the help of vacuum pump. Once this is reached zero, then we close this outlet and take the vacuum pump tube out. Now what we do is we fill a football bladder with hydrogen. Hydrogen cylinder is there with us. This hydrogen cylinder, it is connected with the football bladder. The hydrogen is put inside the bladder, it is full, and that hydrogen bladder is to be put here, and this knob is then opened. Once this is opened, all the hydrogen inside the bladder will go inside. We keep it like this for about 20 to 30 minutes, so that when the reaction is taking place inside, hydrogen and whatever remaining oxygen is there after vacuum, there is some amount of oxygen still left. So that left out oxygen and the hydrogen, they will combine in the presence of catalyst palladium, which is there underneath this lid, and water will be formed. So some amount of hydrogen will be consumed like this, and whatever is consumed, the bladder refills the whole thing. After 20 to 30 minutes, when we see the reaction has completed, we close this tap also, and this is now ready for incubation at 37 degrees Celsius for about 24 hours. Sometimes some anaerobic bacteria, they take a longer time to grow. So depending on the bacterium, if suppose there is no growth, then keep it for 2 to 3 days further. This, I was talking about this methylene blue will be blue in color if there is oxygen inside. If there is absolutely no oxygen inside, then it will be colorless. So you can say that oxygenated methylene blue is blue in color, whereas unoxygenated is or reduced methylene blue will be just like water. This is then indicator to know what are the conditions inside the jar. This is what we do with the Macintosh Fildes jar. The another method is, like I told you, oxygen displacement method. This is done with the help of a gas pack jar. This is the gas pack jar. Similarly, you can see the basket inside. Once these plates are inoculated, they are put inside this basket just like the anaerobic jar. And after that, let me show it to you. This is the basket. This was the lid. After putting the various these plates, inoculated plates inside it, this is the gas pack. This gas pack contains certain compounds, certain chemical compounds. We nick it. With the help of scissors, we make a nick and put water inside it. Once we do it, we place it inside the thing and put the lid on. What is happening now is these compounds, when they react with water, they produce they produce hydrogen and carbon dioxide. And also there is a catalyst inside. So when hydrogen is produced, it combines with the oxygen present in the jar and makes water in the presence of catalyst. Carbon dioxide is also needed for the growth of certain bacteria. So 5 to 10% carbon dioxide atmosphere is also created in this jar. So that is how, and after that we put it in the incubator, similarly just like Macintosh. So this was oxygen displacement method with the help of a gas pack jar. This is gas pack jar. This is vacuum method. This is called Macintosh Fildes jar. Another method I told you is by the reducing agents. Here we have this Robertson's cooked meat medium. You can see the cooked meat inside this. This cooked meat acts as a reducing agent, and when we inoculate this medium with the specimen, this acts as a reducing agent, and the oxygen is there is no oxygen inside the medium, and the medium has become anaerobic. There are no oxygen in this. So this is another method by reducing agents. So these are the three methods we use in a normal laboratory for the culture of anaerobic bacteria. Method of doing anaerobic culture is by the vacuum method, and this vacuum method is done in a jar called Macintosh Fildes jar, which is this. The Macintosh Fildes jar is actually a metal jar having a airtight lid on top of this. This is the lid, and this is secured in its position with the help of this clamp. This is the clamp we use. On the top of this lid there are certain things. One is the meter, which shows how much pressure is inside, to see whether there is this gauge tells us whether the vacuum has been created. Once the needle goes to zero, then we know that this is the vacuum has been created. There are two knobs here. One is inlet and the other one is called outlet. Besides that, there is another outlet here which is connected with a tube containing methyl blue. I will tell you what is the purpose of this later. So once you take the lid out, underneath it there is a sachet. This is the sachet, and this sachet contains palladium pellets, and this palladium it acts as a catalyst for the conversion of hydrogen and oxygen into water. So this is a cold catalyst, meaning that it does not require to be heated. Previously, in the old Macintosh jars, there used to be electrical knobs here so that the current could be passed and this sachet could be heated. But these days, we do not have this thing. We have palladium catalyst now. Inside the jar, there is a basket like this in which the various plates are put. So once we have this specimen for the culture of anaerobic bacteria, the suitable specimen for this is collected usually by syringe and needle like this, and once it is collected, the needle part has to be stuck by a rubber cork. And this is now the specimen which has to be inoculated for anaerobic culture. And what we do is we place a drop of this on blood agar like this. ये क्या हुआ?